All right. Yeah, thanks for having me. I presented a very similar topic or presentation just a few weeks ago at the ULF. Some of you might have heard about this, but it's basically talking about this one drug that has been now in trial for a long time. It's been probably 7 years or so, or perhaps 8 years. Just in terms of disclosures, have served in the past as a PI for this company that owns Leriglitazone. They also help making slides. You know, you're old when people stop helping you making slides. Having said that, I spent probably less than 1% of my effort on Leriglitazone. So the challenge that we have in adrenoleukodystrophy is that we have various phenotypes, and while we have hematopoietic stem cell transplantation and Skysona, there is still no treatment for the adult population, and we have the adult males with AMN, adrenomyeloneuropathy. We have adult females that have a very similar phenotype, but a little slower, also adrenomyeloneuropathy. And about 20, perhaps up to 50% of all adults develop adult cerebral ALD, which is a relatively rapid, progressive, demyelinating inflammatory brain disease. And then we have also the pediatric population that is too late for bone marrow transplant, which is still happening, unfortunately, more than it should. And there's no effective treatment for that population neither. There's also, of course, a concern that neither gene therapy nor bone marrow transplant, and I apologize to all the transplanters here, these therapies suck, okay? You're hurting your patients. So anyway, so it would be nice to have something a little less painful. So one of the small molecules that has been around is a derivative of pioglitazone. It's called Leriglitazone. This is based on some very basic research done in Aurora Pujol's lab about 15 years ago, showing that this drug, these category of drugs can serve as peroxisome proliferators. And so this PPAR gamma induction results in activation of several cellular pathways that sort of have an antioxidant effect, an anti-inflammatory effect, and increased peroxisomes. However, none of the studies have demonstrated a reduction of very long-chain fatty acids, which is the sort of the plasma biomarker to reflect peroxisome function. So how it works exactly in ALD is thought to be multifaceted. It's sort of a dirty drug. And the first human trial was done in AMN, and the study is completed and published now a couple of years ago. This was called the ADVANCE trial, and it was a phase 2/3 study in adult patients with ALD. And this was basically all males, and it included men who had essentially either normal MRIs or had a non-gadolinium enhancing brain MRI that appeared to be stable. And it was a placebo-controlled trial with a 2-to-1 ratio of enrollment. And the primary outcome measure of this study is called a 6-minute walk test, which is probably a terrible outcome measure because it's so dependent on your level of motivation. There is potentially day-to-day variability. And then also, you know, when you progress from walking to using a cane, you might actually become faster because you're more stable. And then when you go from a cane to a walker, you may not be able to count it anymore, and then you end up in a wheelchair. So it's not a great outcome measure. There were secondary outcome measures included in that study, including one important variable called the sway. And so that is basically having a patient standing on the force plate and see how much they move from side to side. The measurement is in a few millimeters. And so the FDA, when first encountered this question whether it could be used as an outcome measure, they said exactly this one sentence that I remember saying: we are not going to approve a drug based on a knee-jerk response, so we are also not going to approve a drug just because somebody's sway improved by 3 millimeters. So you need to show us that it's clinically meaningful. And at the time the study was completed, we had not yet demonstrated that the sway is meaningful. And the bottom line is that the primary outcome measure of the study failed. So when you took the entire cohort that was treated compared to the placebo, there was no beneficial effect. Interesting, in the sway cohort, when you looked at the sway, there was actually a significant difference. And that difference was more pronounced when you looked at patients who were at earlier stages of the disease. But overall, the FDA thought that this was a negative study. As a side observation in that study, it was noted that the individuals who were on the drug as opposed to the placebo, and remember, there were 1/3 of the population was on placebo, 2/3 was on the drug, had a lower chance of developing either new active demyelinating lesions or show progression of existing brain lesions. And also, there appeared to be less of a change in neurofilament light chain. And so this was a coincidental sort of finding. It was too small, certainly statistically, and even in a post-hoc to try to claim approval from that level of data. We talk about the 6 patients or so in this cohort. But it was very notable that, hey, it looks like it's doing something to cerebral ALD. And so this resulted in the establishment of 3 other trials. One of them is the Nexus study that is just completed. Is it published yet, Eric, or almost? Almost. Yeah, the Nexus study, still not published yet. Okay. The final is not yet, yeah. So this is a 90-week long trial where this was an open label study. And here the approach was taken to look at the young boys who have tiny demyelinating lesions that Eric showed you earlier today, those small lesions very early on in the disease, before they go basically for bone marrow transplant, and treat these kids with the drug and see if we can essentially delay the bone marrow transplant. And it's difficult to do a study like that because they started enrolling patients who had gadolinium enhancing tiny lesions and non-gadolinium enhancing tiny lesions. And as you just learned earlier again from Eric, it takes about 4 to 6 months or so for those tiny lesions potentially to show enhancement. Well, we don't know exactly. And when you look at these as 2 separate populations, the results weren't super impressive. When you looked overall, it looked like there was actually a significant effect again on the drug on decreasing the likelihood of cerebral progression compared to historical controls. You can see also from a lesion volume perspective here, the gray are historical controls meaning ALD cases that were not treated. And then in this open label study, the blue cohort. And so there seemed to be a difference. When you look at the population that was gadolinium negative, they looked great. They did very well on the drug. When you look at the population that was gadolinium positive, there is a question whether it seems like the progression is slower, certainly than you saw those rapid volume increases that Eric showed, and here it looks a little less steep on the right side. But it's a small study, I would say. And so, as expected, these are early lesions, so nobody developed any worsening neurological disability. The data showed also that this drug is quite safe. We have understood now its side effects in these populations. It causes some weight gain, some periorbital edema. So you can, even though it's placebo, many times you can tell who's on the drug. And so far, no serious adverse events aside from a couple of patients that had liver issues, but they're likely not related to the drug. So then, in the same time, there was also a compassionate use study that is done in France. This is really compassionate use. It's not a real study. And it was done in adult cerebral ALD cases that basically were helpless. And even when you look at that cohort, and I had a couple of my own patients sent to that study, there was definitely some effect in some individuals. The gadolinium enhancement would go away, the lesion would stop progressing, which again, there is a problem here because it's compassionate use. There's no placebo, and the disease course in adult cerebral ALD is not very well characterized yet. So the FDA said none of this is enough. We need a full placebo-controlled trial to see if this is actually working or not, this drug. So currently we have had an ongoing study called the CALIX study. That's a multinational study, which is a 1-to-1 placebo-controlled trial of adult patients with cerebral ALD who get treated up to 36 months. After 18 months, there's an interim analysis that will be done. And the outcome measure that the FDA agreed to was survival. So that was a tough conversation. We had a lot of back and forth. We were trying to use the AACS, which is an adult ALD scale, clinical scale. They didn't think that was good enough. And so ultimately, it's either survival. So you either look at somebody who dies as a negative outcome or who's fully ventilatory dependent, essentially in the vegetative state. So this study is currently still ongoing. You basically have to be an adult male who's either choosing not to go for bone marrow transplant or is for whatever reason deemed to not be a bone marrow transplant candidate. I'm not super excited, to be honest, about adult bone marrow transplant in ALD. I know Paul disagrees with me. Again, we're going to be looking at survival and then a bunch of other secondary outcome measures. This study is still enrolling. We have had difficulty with enrollment, so the company was forced to expand sites. So there's many sites now across essentially the American continents, Europe, and then most recently also India coming on board. So if you have any patients, please let us know or refer them to clinicaltrials.com or to the Calyx. They can just Google Calyx. That would be it. Thank you.